Some of the most important biological macromolecules are the proteins. Proteins are large molecules made by building blocks, smaller molecules, called amino acids. Proteins consist of long chains of 50 or more amino acids held together by covalent bonds and folded into a complex structure. Functions of proteins are varied. Some of the most important functions found in some proteins is that of being enzymes. Enzymes are biological catalysts found in cells that speed up chemical reactions. Other proteins are involved in structures that are used by cell movement, like flagella or cilia. Other proteins are found inside the nucleus of eukaryotic cells or the nucleoid of prokaryotic cells, binding to DNA, regulating gene expression. Yet other proteins are involved in transport of molecules in and out of the cell. The shape of a protein determines its function and its characteristics. Here's a depiction of different protein shapes. Each of these proteins have a different function, thus a different shape. Proteins are made of smaller building blocks called amino acid. There are 20 naturally occurring amino acids in the living world. Each of the 20 amino acids have a similar chemical group. Each of the 20 will have a central carbon attached to an amino group on one side and a carboxyl group on another side. The central carbon will also be attached to what is called an R group or side chain. This will be the part of the molecule that will differ among the 20 amino acids. Here is a depiction of 5 of the 20 amino acids. Notice the R group in glycine being different from the R group in phenylalanine or cysteine. It is the R groups that give each amino acid its individual chemical characteristics. Amino acids are brought together by covalent bonds called a peptide bond. A peptide bond is the result of a dehydration synthesis in which the carboxyl group of one amino acid reacts with the amino group of another amino acid. With the removal of water, a covalent bond is created between the two amino acids. This covalent bond is called a peptide bond. This figure shows you 3 amino acids bonded together by 2 peptide bonds. This chain of 3 amino acids do not constitute a protein. This is called a polypeptide. Polypeptides are short chains of amino acids. Protein function is linked to its shape or structure. As proteins are made by ribosomes into long chains of amino acids, the long chains are folded into 3-dimensional structures. Those proteins are said to have primary structures, secondary structures, and tertiary structures. Some large proteins may even have a quaternary structure. The primary structure of a protein is its sequence of amino acids held together by peptide bonds. The strong peptide bonds that hold together the sequence of amino acids constitute the primary structure of a protein. Adjacent amino acids quickly react with each other, twisting the chain into a helix or folding the chain into a pleated sheet. Reactions between adjacent amino acids form weak chemical bonds, hydrogen and ionic bonds, which are the bonds that hold together the helix and the pleated sheets, which constitute the secondary structure of a protein. Amino acids from helices and pleated sheets react with each other, again forming weak ionic and hydrogen bonds and twisting the protein into a three-dimensional shaped structure. This constitutes the tertiary structure of a protein. Some proteins consist of two tertiary structures in which amino acids from each form weak hydrogen and ionic bonds, bringing together two or more tertiary structures. These proteins are said to have a quaternary structure. Remember, the primary structure of a protein consists of the amino acid sequence formed by strong covalent bonds. Secondary, tertiary, and quaternary structures of a protein consist of weak ionic and hydrogen bonds. Proteins are very sensitive to their environment. Extreme heat or extreme pH will break these weak hydrogen and ionic bonds, destroying the tertiary and secondary structure of a protein and quickly bringing it back to its primary structure. This process is called denaturation. So proteins can be denatured by extreme changes in their environment back into their primary structures. Proteins are also antigenic molecules. Remember that an antigen is a molecule that triggers the formation of antibodies in an immune system cell. The presence of foreign proteins in, for example, a bacterial cell will trigger an immune system cell into producing antibodies that will bind to the protein that trigger the production of such antibodies. So, proteins are said to be antigenic because the presence of proteins trigger the formation of antibodies in immune system cells. You now should be able to answer the following questions. What are the building blocks of proteins? What determines the function of a protein? How can a protein be denatured? What are catalysts? What are enzymes? What are polypeptides? What type of covalent bond holds together amino acids? What cellular structures are made of proteins?